Everyone. Welcome to the seminar. So hope you all survived the heavy snow today. So today I'm very glad to introduce today's speaker, Dr. Lesley Schumacher. Schumacher, right? Did I pronounce the right correctly?
Schumacher.
Schumacher. Okay. Yep. Sorry, Dr. Schumacher. And so basically, Dr. Schumacher got her bachelor and master degree from the Michigan State University. During her master degree, she worked on the soybean cyst nematode and the root lesion nematode associated with soybean in Michigan. So after that, she moved to the Florida University and did her PhD in the Department of Entomology and Nematology. It's actually one of the few departments in the US with nematology in the department name, so. And for her PhD, she worked on a different nematode, which is the Reniform nematode management in the peanut and cotton system. Shortly after her PhD, she can move back to the Midwest, worked as a research plant pathologist at the USDA-ARS, based at Jackson, Tennessee. So her current research program focused on the soybean cyst nematode resistance and the plant-parasitic nematode management. So with that, Lesley, the floor is all yours, so you can start your talk. Thank you.
Thank you. Thank you for the opportunity to speak with you guys today. I wish I could be there in person, and hopefully one day soon we can be. So I'm just going to make sure, start this here. I'm assuming everyone can see that. I know those famous words everyone likes to say.
Yeah, you're good.
Okay. So as you can probably tell from my Midwest accent, I'm originally from Wisconsin, and then my family moved to Michigan. So I'm very familiar with the area in and around Indiana, as I was just a stone's throw away from there. And between my master's and PhD, I actually was a dairy farmer and a maple syrup producer while working in various positions at Michigan State University. So before I decided to go back to the University of Florida, I actually was the academic advisor for the Department of Forestry at Michigan State. So I have sort of a wide variety of experiences in my background. But today I want to talk to you about this concept of utilitarianism in nematology, and how we can find effective solutions to solve crop production challenges. And just to give you a little bit of overview about our work site. So we are part of the Crop Genetics Research Unit, which is based in Stoneville, Mississippi, and it's part of the southeast area of the USDA. But we have a small group of scientists and technicians and some field workers that work in Jackson, Tennessee. So that's where I'm going to be talking about today. Now, why do we care about nematology? Why is nematology important? And why do I personally love nematology? 4 out of every 5 animals on the planet are nematodes. And this is because they can colonize both marine environments and terrestrial environments. And if you were to go outside and collect a small container of soil and know how to extract the creatures that live within there, you're going to find things like small insects or collembola, fungi, bacteria, and of course, nematodes. And what nematodes are is a diverse group of roundworms. In one handful of soil, there can be hundreds, if not thousands, of these creatures. They can also feed on animals, bacteria, fungi, and plants. When we study plant nematology, we're focusing specifically on those nematodes that feed on plant roots and can inhibit crop yield and growth. Pretty much all plants are affected by plant nematodes, but today I want to focus in on soybeans and cotton in particular. You can think of nematodes as both parasitic and beneficial, or simply speaking, bad and good nematodes. Our bad nematodes we have to manage them in order to achieve our desired crop yields. And examples of those are cyst nematodes, Reniform, and root knot. I'm going to be talking about two of those in detail today. We also have beneficial nematodes or good nematodes, also known as non-targets. And these are good soil health indicators and can actually contribute to soil fertility. And this includes bacteriivores, fungivores, omnivores, and predators. And I don't know if anyone can see this book right here, but this is a concept that is not new. And this is a book called Nematodes as Environmental Indicators. So studying populations of these beneficial nematodes can tell us about the health of our ecosystem. Jumping back to our plant parasites, this is what these look like up close. So we have a nematode's head embedded in a plant cell, and they feed on plants by using this stylet, which is acting sort of as a needle, and that's what it's using to feed on root cells. That brings me to today's topic of utilitarianism nematology. That's just a fancy way of saying how we want to find the most effective solutions that are going to solve our biggest problems. You might recall from your middle school or high school literature classes this concept of utilitarianism, and it's a philosophical doctrine that the actions are right if they are useful or for the benefit of a majority. And another way of looking at that is actions should be chosen based on their likely results and maximized utility. Today, I want to explore this concept as it relates to nematology. And we're going to do that by talking about two main plant parasitic nematodes. The first is soybean cyst nematode, or Heterodera glycines. And here I have two soybean root systems, and on the top, you can see a white arrow pointing to a small white dot. And that's actually the female worm as she has burst through the root surface. Compare that to the root system on the bottom, where there's a green arrow pointing to a nitrogen fixing rhizobium nodule. That's just a size example for you to see. The other nematode I want to discuss is Reniform nematode, or Rhodolenculus reniformis. And this is what a female looks like as she has burst through a cotton root in this example. To get us started, some fun facts and history about nematology as it relates to these two nematodes. So Reniform nematode was first found in Hawaii in 1931, infecting cowpea. And this was a really important discovery. It actually turned out to be a significant pathogen of pineapple, and it led to the use and discovery of fumigant nematicides, which basically gave birth to plant nematology. In this case, it showed that nematodes were the causal agent of damage to the plant and reduced the yield of the pineapples. This particular nematode can feed on more than 300 host plants, and it gets that name Reniform because of its kidney-like shape, as you can see in this image here. Soybean cyst nematode was first observed on soybeans in Japan in 1915, but in the US, it wasn't found until 1954 in North Carolina. The genus name Heterodera means different skin, and you'll see that in the next few slides as we begin to look closer at what that cyst looks like. The disease it caused was originally known as yellow dwarf disease because it caused the soybean plants to become stunted and yellow. Even though we like to talk about soybean cyst nematode in the realm of soybeans, it actually can feed on more than 500 host plants. Since its introduction to the US, it has since spread to all of these areas in red and continues to do so on an annual basis. While I was at Michigan State, I was actually able to fill in 2 more counties while I was doing a survey for my master's project. And results from that survey, I found that about 50% of the fields that we surveyed were positive for this nematode. It's considered the #1 limiting yield factor of soybean in the US, and it causes around $1.5 billion in annual losses. It can also interact with other pathogens like fungal pathogens, sudden death syndrome. The main issue that we're having is that resistance is losing its effectiveness. The main source that we have been using on around 95% of our infested acreage is PI 88788. And after a while, we've been selecting for population densities to become resistant to this resistant source. Therefore, we have to revise our management approaches. And this is because this nematode can survive for up to 12 years without a host. And this is why. If we go out into our field around July, dig up our soybean plant, and we see all these white dots, we know that we have a problem. Once those females mature, the males in the soil will mate with them, and their bodies basically become this egg-filled protective cyst. Those cysts will then be distributed into the soil, and there can be around 250 eggs inside each cyst. So the populations can build really quickly. In each one of those eggs is a juvenile, and this is what's actually going to hatch out and be searching for a soybean plant to infect. Switching gears to Reniform nematode, this is more of a problem in the cotton belt states, which Tennessee is considered. And this image here shows the thousands of cotton bales that are lost per state due to this nematode. It amounts to around $252 million in revenue loss. And not only is it a pathogen of cotton, but it also is a pathogen of soybeans and can cause a 2 to 60% reduction in yield. The way that this damage manifests is in waves of uneven cotton growth. We also can get stunting and yellowing and wilting as well. I want to talk briefly about 2 other plant parasitic nematodes that we're going to discuss in a few slides. And that is the spiral nematode or Helicotylenchus dihystera. This is what that particular nematode looks like, named very appropriately. This is an ectoparasite. So that means as it's feeding, its body is going to remain outside of the root. It has a very wide host range, which includes cotton, peanuts, and soybean. However, any damage that it causes is considered minor and might be in the form of root pruning. Another common plant parasite is the ring nematode or Mesocrichinema ornatum. This is what this looks like. It's a very heavily annulated nematode and also an ectoparasite, and it's considered sluggish. And that will become more apparent in just a few slides. This nematode doesn't move a lot, and until a certain extraction method was developed, it wasn't even detected in fields. It also has a wide host range that includes peanut. And when it damages peanut, it manifests in the form of smaller than average pods. Now, let's switch gears and talk about our non-target soil organisms, also known as our beneficials. When we are extracting nematodes for a plant parasitic diagnosis, we're actually getting several other creatures in those samples. And these are free-living nematodes, so they're not parasites. And they're feeding on bacteria, fungi, they're feeding on a wide variety of sources such as the omnivores. And then we also have predators that can feed on other nematodes or other soil fauna. And this is what each one of these looks like. We identify these morphologically, but we can use molecular tools as well. And a lot of times when I'm counting these in my samples, I'm looking at the mouth part in order to determine what they are and what they feed on. So we've got the bacteriivore on the left, the fungivore, the omnivore, and the predator that has that big, wide mouth, and you can imagine it can easily slurp up another nematode. We also have things like rotifers, oligochaetes, mites, collembola, and everyone's favorite tardigrades or water bears. This is just what each one of these looks like. Collectively, all of these creatures are responsible for nutrient cycling, and they can be sensitive to environmental disturbance. So by looking at their population densities in relation to our plant parasites, we can learn a lot about the soil health. That brings me to our challenges that we are facing. The first one is nematode diagnosis and sampling. How can we improve this process? The second challenge is with the elimination of methylbromide as a soil fumigant, we are left with these new non-fumigant nematicides. And we want to know, how do they perform and what are their impacts on those soil fauna? Our third challenge is crop rotation. Specifically, how does it affect nematodes and yield? Our fourth challenge is concerning soil health, a really hot topic nowadays. In our realm, we want to know how cover crops, the burn down timing of those cover crops, as well as how our seed treatments are affecting the soil fauna. And our final challenge for today is SCN resistance. Specifically, we know that PI 88788 is losing the battle. How can we help in this process? I'm going to share with you results from the North in Michigan, the Mid-South here in Tennessee, and the South in Florida. So let's get started. And this is my nematode management wheel. At the north end of the wheel, we have our chemical management in the form of fumigant and non-fumigant nematicides. And at the south end of the wheel, we have cultural management like cover crops, crop rotation, resistant cultivars, and sanitation. In the middle there, we see the first step is management in the form of sampling and diagnosis. So we're going to start there. How can we improve this process? Let me walk you through what we are currently doing. So we go out to our field, we have our soil probe, and normally we like to sample in a zigzag pattern because we know that nematodes have a patchy distribution. But when we do that, we might be missing large quantities of, for instance, SCN cysts, just by being a little bit off with our soil probe. So that comes to the question, okay, how many samples should I take? When should I take the samples? All of these steps can be improved on. And another way we can look at this is during the growing season. So we can go to our field and we can say, hey, something looks like it's going on in this particular area. We'll go out, we'll dig up those plants, take them back to the lab, observe the roots and see if we have any symptoms of infection. We can also evaluate our field and yield history to help us with our diagnosis. And finally, continue to have our soil and roots processed for nematodes throughout the growing season, ultimately winding up at the microscope, which is what we have been doing now. That leads us to one of our lab's key goals in the future, which is to improve our methods in nematology. And we are going to do that by testing novel equipment for its application to plant nematology. Our methods in this field are very old. So we have the Behrman method that was developed in 1917. We have a blender method developed in the 40s, and our tried and true method, sucrose centrifugation, developed in the 60s. And you might be thinking, well, if it's not broke, don't fix it. Well, we can still improve upon these processes and make it easier for us to perform our work, which is screening and efficiency of getting these nematodes out of the soil. And this is because the method matters. We can inform our management decisions based on the extraction method that we use. Here's some data from a recently published manuscript. And this is looking at the total number of Reniform nematode life stages per gram of cotton root tissue. And when I mean life stages, I'm talking about eggs and the vermiform or worm-shaped stages. Looking at these different methods, those three methods that I mentioned, the Behrman, blender, and mist chamber, we can see that the blender method gave us the best results. And that's important because we can actually use this concept going forward to detect treatment differences within the field based on the method that we are using. And let me get into that. First, I want to talk about the long-term rotation study that we used for this work. Coming from the north, I had never seen peanut or cotton. So just in case you haven't, this is what these look like. This is a row of peanut and a row of cotton. In the south, our conventional rotation is peanut followed by 2 years of cotton. We also have this alternative rotation called a sod-based rotation that uses 2 years of a perennial grass, then followed by 1 year of peanut and cotton. And this rotation was developed because it had great agronomic benefits. It increased water infiltration and reduced pathogen density. It's kind of unique to North Florida. We had a lot of cattle in North Florida, so growers could easily transition their ground to graze their cattle for several years. Then they could go back and putting it into production and would get really fantastic yields in either their peanuts or cotton. So that's why this rotation was developed. And this was a long-term study that we conducted in Quincy, Florida. It was actually a split plot. I had irrigation as my whole plot factor, crop phase as the subplot, and fluopyram nematicide as the subplot at 18 ounces per acre, which we applied in furrow at planting. This research was conducted from 2017 to 2019. A typical field replicate looked like this. So we have our 4 phases of that sod-based rotation shown in this green color here. And then in the gold color are the 3 phases of our conventional rotation. So cotton 1 means first year of conventional cotton, and cotton 2 is our second year of conventional cotton. Drilling down a little bit further, I'm going to just discuss these 3 phases. So 1 from the alternative phase and 2 from the conventional. So what I wanted to do is assess those 3 common root extraction methods for their utility in recovering the reniform nematode life stages from the cotton roots. The blender method is the image on the right. The modified Behrman is the image in the middle. And on the left is what we call a mist chamber. So these are common methods that are used by diagnostic labs, and that's why I wanted to run this trial. Specifically, which methods work for reniform? I would go out to my plots. I had 42 plots. I would randomly dig up 9 cotton plants, and then I subjected 3 to the blender, 3 to modified Behrman, and 3 to mist chamber for each plot. Looking at this particular response variable, our vermiform or our worm-shaped stages of the reniform nematode, here is what we found. So this is showing the population density per gram of cotton root. And then we have our 3 different methods here on the x-axis. And here are those 3 cotton phases. So sod-based cotton, cotton 1, and cotton 2. This is for 2017. So we say, okay, all 3 methods gave us the same statistical results. That's great. Let's see what happens in 2018. All of a sudden, we're starting to see some differences with the mist chamber extraction method. And we can see that now within the conventional phase, we're seeing statistical differences. Let's look now at the reniform nematode eggs as our response variable. Again, this is in 2017 and 2018. And while we are still seeing some treatment differences using the blender method, the Behrman and mist chamber methods did not recover any eggs. So we would not have been able to detect them if we were a diagnostic lab using this. What this all boils down to is active versus passive extraction methods. The blender method worked well for extracting reniform eggs because this is a passive method, meaning that anything that's in those root tissues is going to be liberated during the blending process. Versus those other 2 methods are active, and it relies on the nematode hatching from the egg stage crawling through a filter to be ultimately collected in the bottom of either the bowl or the flask. And recall back to the ring nematode, me calling it sluggish. This is why this was important. This nematode could not be detected using those active methods. It was only in the 1960s when the sucrose method was developed when people could actually determine that ring nematode was actually a problem. This just goes to show how important it is to understand the biology of your target nematode and also the limitations of different methods. And it's really important to select the appropriate method. Otherwise, you might misrepresent treatment effects from the field and ultimately change your management decisions that you are going to pass on to your growers. Let's move to a different part of our management wheel now, and I want to talk about nematicide and crop rotation. The whole goal of using a nematicide is to kill the nematodes. And the non-fumigant that we chose was fluopyram, which also happens to be a fungicide. This is what it looked like when we were applying that in furrow at planting. And I want to talk a little bit more about crop rotation, which the whole goal of that is to starve the nematodes by rotating them away from their food source. And as I've kind of already discussed, this is the rotation we used: cotton, peanut, and the bahia grass. So let's look at the results from these management strategies. So here, once again, we have our population density of reniform nematodes per 100 cubic centimeters of soil. And on the x-axis, we have our plots with no nematicide and nematicide. You can see that statistically, there was no difference in the reniform nematode population densities between plots with and without nematicide, even though numerically we might see a trend there. What about those non-target or beneficial nematodes? Well, we also saw no difference in population densities between plots with and without nematicide. And this is something that chemical companies are really interested in going forward. They want to know if there's any non-target effects. Well, if I collectively looked at free-living nematodes, I didn't see any. But when I separate them out into their specific feeding groups, we see a different story. And we can see that in all three of these sampling dates, the omnivores were negatively affected by nematicide. This is actually nothing new. Others have documented this same trend where omnivores are particularly sensitive to chemicals. But other nematodes might not be so sensitive to chemicals. And this is the case with the spiral nematode that I encountered in my field trial. These results have been published in the Journal of Nematology, so I welcome you to take a look at that if you are interested. And here are all 6 sampling dates of my study. And in 5 of those 6 dates, the population densities of spiral nematode were actually significantly greater in the plots with nematicide. Once again, this is supporting a trend that others have observed. This was observed in turfgrass studies as well. And it seems like nematodes in the family Hoplolimidae, which includes spiral nematodes as well as lance nematodes, are both kind of having the same trend. Something interesting to consider in the future. Let's move on to what a grower wants to see. What is happening in terms of yield? So we can see here that our plots with nematicide actually did give us a significantly greater lint yield than our no nematicide plots. And that amounted to around 120 pounds per acre increase. If we do a little bit of economics on that and figure 75 cents per pound of lint, we're getting a $90 per acre increase using the fluopyram. If we subtract the product cost from that, the grower can expect to see around $50 per acre yield increase using that product. And this is perhaps because it was controlling fungal pathogens that might have been more detrimental to the yield than the nematodes. In summary, for our chemical management, in our study, nematicide did not manage reniform nematode. However, it did negatively affect the population densities of omnivores. We did see a return on investment, but let's see how crop rotation performed. So once again, we've got our reniform nematodes on the y-axis, and now we have those 3 phases of the cotton on the x-axis. We can see that the reniform nematode population densities were greater in the conventional cotton than in our sod-based cotton phases. And looking at our beneficials, those 2 were also greatest in one of the conventional phases in the second year conventional cotton. When we look at yield, we can see that our sod-based cotton had significantly greater yield than any of our conventional phases. And that actually worked out to be around 355 pounds per acre. So when we run the economics on that, we are seeing a $266 per acre yield increase with the sod-based cotton over our second year conventional cotton. Going back briefly to our population densities of Reniform, I wanted to show you throughout all of the sampling dates what these looked like. So we have the earliest sampling date to our final and our 3 cotton phases here. And we see that indeed in every single sampling, we had greater Reniform nematodes in the conventional cotton plots. Now, keep this in mind going forward, because when we look at another nematode, the ring nematode, we're seeing the exact opposite trend, that the ring nematode populations were greater in the sod-based cotton than our conventional cotton plots. In summary, for crop rotation, this actually managed our Reniform nematode better than chemical, and our beneficial nematodes were still the greatest in the conventional rotation. Now, the whole point of talking about utilitarianism in nematology is we saw that managing one nematode increased the population density of another. However, because ring nematode is not a significant threat to cotton production, we're not really concerned that we increased that nematode. So we want to find the biggest solutions that are going to solve our most pressing problems. Moving now to a different part of our wheel, now we're going to talk about cover crops, and this is current work that we are doing in our lab. Cover crops are a really hot topic right now as well, and this is because they may suppress pathogens and also help improve soil health. We can use a variety of mixes. We can use cereals, brassicas, legumes, or a combination of them. And we plant them in the fall after we harvest our cash crop, so soybeans, for instance. Then we burn them down in the spring before we plant the next year's cash crop. This is an aerial image of the trial that we conducted last year, and you can see the fallow strips and the cover crop strips here. This is actually a 3-way factorial, and our first factor was cover crop treatment. So we did a 5-way mix, then we did a 6-way mix with basically the addition of a brassica, and our control was a winter fallow. Our second factor was burn down timing, which we did 3 weeks before planting, considered early burn down, and then our late burn down was at planting. Our third factor was seed treatment, and we had insecticide only, fungicide only, a combination of the 2, and then a 3-way treatment with the addition of a nematicide. Our objective in this study was to determine the potential benefits and or disadvantages of using cover crops as they relate to plant-parasitic nematodes and the soil faunal community in soybeans. I also want to talk about this metric that's not commonly assessed, and that is eggs per cyst. This is a measure of fecundity, and if we can calculate the number of eggs per cyst, it'll help us determine the utility of those soybean cyst nematode resistance genes. So if the nematodes are not able to produce as many eggs, we're able to reduce the inoculum for the following growing season. The way we do this is by first assaying the number of cysts per soil sample using this piece of equipment called an elutriator. And this is actually floating the cysts out of the soil, uses a lot of water. And once we float those cysts, we can count the number of cysts, crush them using a modified drill press, which releases the eggs over these nested sieves. Then we can count and using simple math, determine the number of eggs per cyst. So keep this in mind as we talk about the results. These are just preliminary data, and this is from summer 2021. We have our eggs per cyst and our seed treatment. We had a significant effect of seed treatment on this particular response variable. And interestingly, we see that our fecundity rate here is greatest in the three-way seed treatment plots. When we move to our burn down timing factor and in regards to our different soil organisms, so we have our population densities here and the different soil organisms, we're seeing a really consistent trend. And that trend is that in the early burn down timing, we were always seeing significantly greater population densities of these creatures versus the late burn down timing. In summary, for our cover crop management, so far we have not had any significant effects on our response variables, but these, as I said, these data are preliminary and we are still working through them. We saw that seed treatment did affect the SCN fecundity. And finally, burn down timings may negatively impact our beneficial soil organisms. Moving to the last topic for our management wheel is resistance. And now I'm going to switch gears and talk really heavily about soybean cyst nematode and what HG types are and what we can learn from the field using them. HG types, that's just a fancy way of saying heteroteroglycines, and this is referring to population densities of soybean cyst nematodes that can reproduce on our resistant PI lines. This system replaced the race system, and we use these 7 sources of resistance in order to tell us about those population densities. For instance, a really common HG type is 257, which was known as race 5. And that means that that particular nematode can reproduce on PI 88788, PI 209332, and PI 548316. So growers will not be able to use that common source of resistance, PI 88788, and knock down the population densities of an HG type 257 nematode. And what I wanted to know was, what happens when we mix different sources of resistance together in the planter? We did just that back at Michigan State in 2010. Our plots were 4 rows each, 250 feet long, and in Michigan we use a lot of tillage. And I've categorized these population densities based on these colors. So in red, we have greater than 3,000 eggs and juveniles per 100 cc's of soil, ranging all the way down to category 1, which is no SCN detected. The top graphic here is showing our initial population densities. So that's in the spring prior to planting. That's what was there for the plants to have to deal with as they went through the growing season. And the bottom is our final population densities. And here I have a susceptible variety, a mixture, which includes all of the different sources of resistance, including that susceptible, 1 88788, 2 Pekings, and 1 437654. You can see that over the growing season, in this mixed plot, we were able to pretty effectively reduce the population densities from the red down to more manageable levels. Just to give you an idea, relating back to that fecundity measure, when we are dealing with sources of resistance, the PI 437654 by far gives us the smallest cysts with the fewest number of eggs. versus our 88788 produces the largest cysts with the greatest number of eggs. And that's all relative to our susceptible, which obviously produces the greatest number of cysts with the largest number of eggs. Using this concept, I wanted to apply this going forward. And in 5 of these 9 field trials, I didn't want to talk about all of them, we had significant differences between our resistant varieties. But look specifically at that site from 2010, and you can see that even though these yields are still not great, the greatest yield was achieved in that mixed plot. So right now at USDA, we are doing a trial where I'm still processing the data from this past year, but I'll show you the theory behind this. Using what we had learned back at Michigan State, we are mixing different sources of resistance together in the planter at different ratios. And this is what this looks like at the end of the growing season. So in theory, what we're expecting to see, these are the different sources that we mix. So we have a susceptible as well, a Peking, and an 88788. We know that there's multiple generations of these nematodes per growing season, especially so in the warm soils that we have in Tennessee. So we might be seeing something like this. The susceptible has, as we know, going to have higher SCN. Our Peking might have very low or reduce the number, and our 88788 is going to be losing its effectiveness. This is what we can see. And the goal of this, by mixing these in specific ratios, is to make sure that our yield is going to be stable while ensuring the longevity of the resistance genes. This will help us address that PI 88788 problem. Therefore, our objective is to determine which of these ratios is best to both reduce SCN and give us good yield so that the growers can use this going forward in their own fields. Now, getting to the bread and butter of what we do, I'm a new PI in USDA, and I just want to share with you what we are doing on a daily basis. So we do a lot of greenhouse work, and in order to do so, we have to maintain lots of SCN cultures. We keep several HG types active for collaborators, as well as to fulfill internal testing requests, and this is what this looks like in our greenhouse. We have to ensure the identity of those HG types, and we do that by keeping tabs on them using the type testing, but we also reference the race system, and we keep these throughout the greenhouse posted so we know what we have when we are working. We know if we have an HG type 0 or type 257, etc. Annual responsibilities for our lab are to perform the Southern Uniform Soybean tests, as well as county variety trials. We have colleagues that send us around 300 entries to screen annually for the Southern Uniform tests, and several hundred as well for the county variety for our University of Tennessee collaborators. And we do that by screening 3 HG types, 1 seed per pot, 7 reps, and we inoculate those with 2,500 eggs per pot. At the end of the cycle, 30 days, we then rate these based on the number of cysts per root system. And that's what we use when we're writing our reports. Once again, we want to keep the identity of those races in check by running standard susceptibles. Another thing that we're responsible for is germplasm screening. And we either get the germplasm from our collaborators, or we work with the US soybean germplasm collection to test for reaction against soybean cyst nematode. We screen 3 HG types, slightly different protocol where we thin after planting, inoculate with 2,500 eggs per plot, and at the end of 30 days, we actually use water to blast those females from the roots, and then we can count them and even do our fecundity measurement if we so desire. The goal of all of this work is to help identify new sources of resistant germplasm for SCN. overall, I want to say that this concept of utilitarianism, it's a balancing act. We need to be looking at all of the different factors. We cannot just be isolated on our own islands. We have to consider all other pathogens, fungal, bacterial, viral. We need to look at insect pests as well as abiotic factors. And we need to determine what is best for the system and the biggest threat to production so that we can help the growers. To do this, we need to work with other disciplines to find the best solutions that are going to solve the biggest problems. And with that, I thank you for your time and attention, and I'm happy to take any questions that you might have.
Yeah, thank you, Lesley. If you have any questions, please feel free to ask. If not, maybe I can start with a couple of questions.
Okay.
So like, at the very beginning, you mentioned you are working on some novel equipment for the nematode diagnosis. So, but maybe I missed that one. So what's the novel equipment like you mentioned?
So one of the pieces of equipment is used in veterinary diagnostics. And we obtained that last year. Because of the pandemic, we haven't been able to really play around with it a lot yet, but that's part of our upcoming project plan. So I can't share too much details about it other than it's from veterinary parasitology.
Okay, yeah, okay. And the other question you mentioned, for your spiral nematode, you treat them with some like nematicide. So which nematicide did you use?
Fluopyram.
Okay,
It was a vellum total.
Yeah, so the interesting is that it's instead of reduce the nematode population, it's kind of like a, increase the nematode population. Do you think what's the possible like reason for that?
So we're seeing that in a lot of different systems in that particular family of nematodes. I don't know if it's something about that particular family or if we're just reducing competition at that time and that nematode is able to rebound quicker than others. And that could be a potential reason why we're seeing that.
Okay. Does anyone have questions? Any other questions?
I see some questions in the chat here.
Oh, in the chat. Okay.
Somebody asked, how long does fluopyram impact the omnivore nematode populations? I did not do a study where I could see the effects after time. All I can say is that in every single sampling date, it was impacted. So the half-life in the soil of that particular nematicide isn't super long, but I think it might be having, it's having some sort of long-term effect on these omnivores because they're always reduced in those plots. So something's happening there, but I didn't do an actual determination of that. And then somebody asked, for cotton, is a system such as the Bahia grass rotation feasible other than in Florida? I would love for there to be a system, but just because of the nature of the North, and even here in the Mid-South, our grass goes dormant in winter. I don't think it's going to be very effective. And then we have snow cover on top of that, even though, yes, we have a lot of cattle in the North. And I myself would have loved to put that in place at our farm, but I think it's more likely it's going to be just beneficial in Florida. And then somebody asked, has your soybean germplasm screening identified any new sources of resistance? Yes, not mine personally, but our group here in Jackson has identified some new sources, and those have been recently published. And actually, Dr. Zhang works with that. So, yes.
Yeah, thank you very much for your wonderful presentation. So I was looking at I followed when you talked about screening germplasms, and you mentioned that you inoculate the soybean plants with 2,500 eggs, if I'm not mistaken. So I wanted to know if that's an approximate or is a specific number, 2,500, and if it's a specific number, how do you go about to quantify that?
So we do a lot of different inoculum calibrations. We have a counting dish that we count several times to make sure that we have the right number, and then we calibrate that to nematodes per ml. So it's just a lot of balancing, adding more water here, taking water out in order to get it to that final density per ml.
Okay. Yeah, I wasn't just sure how you do that. Yeah, because I'm doing similar work, and so yeah, that sounds interesting. Thank you very much.
You're welcome.
Yeah.
I had a question, Dr. Schumacher. That was a very good talk. I had a question that was also related to the screening of soybean germplasm. I was wondering to what extent does your research program and related research programs incorporate or try to discover nematode resistance to the extent that it exists in kind of wild relatives of soybean, like glycine soya and other related species? Thank you.
So our role is to screen those lines, and then we work with, being part of the crop genetics research unit, we then identify potential candidates, and then we sort of pass that on to our plant breeder. And then they're the ones that are going to be looking to create that elite germplasm from there. So we're just sort of the first step in the process.
Thank you.
Can I ask another question?
Sure.
About the variety mixture study you did?
Mm-hmm.
Many years ago, people were looking at cultivar mixtures in cereals to protect rust resistance genes. And one of the issues was finding cultivars that you could mix in the planter that were similar enough agronomically that it wouldn't cause any issues for harvest date and crop height and so forth. Is that less of an issue with soybean?
Well, what we did, this study has only been in the ground for 1 year. And what we did is we looked very carefully through the seed catalog, and we picked the same maturity group, same very similar agronomic properties in order to make sure that that wouldn't be an issue. So it was kind of a lot of background work on our part to find that. And we had to also find varieties that had nematode resistance with the less frequent sources of resistance, like Peking. So it was a little bit of a challenge, but yeah, we took that into consideration, and it ended up not being a problem for us. So it shows, hopefully we'll see if it has any benefits in the future.
Yeah.
Are there any other questions? Okay, if not, let's thank Lesley, thanks for the talk.
Thank you for having me. And if anyone wants to continue to stay on and chat, I would be happy to do so.
Okay, yeah. Let's